Hello, and welcome to another episode of Max Planck Florida's Neurotransmissions podcast. I'm Joe Schumacher, joined again by my co-host, Dr. Lesley Colgan. How's it going, Lesley?
It's going great. Good to see everyone.
We have a really exciting show today, a big moment for us, a long time in the making. But today we are joined by Professor Dr. Bill Hansson, director of the Max Planck Institute for Chemical Ecology in Jena, Germany. He's the head of the Department of Evolutionary Neuroethology, and he's a former vice president of the Max Planck Society. And Bill Hansson, thank you so much for joining us today.
Thanks for the invitation. It's great to be here.
So we're really excited to have you here today, and you're something of a celebrity around here because of your position in the Max Planck Society as a vice president and your critical role in helping us develop our institute into what it is today in the very early years of our institute. So it's amazing to have you here on the podcast for the first time. And actually, we wanted to start by asking you a little bit about your role as a vice president in the Max Planck Society. So we're this kind of oddball in Max Planck Society in the sense that we're one of the only Max Planck Institute outside of Europe. We're all the way over here in Jupiter, Florida. Why were you paired with us as a vice president? What is it about the structure of the Max Planck Society that had you interfacing with us and other institutes? And then what sort of capacity did you serve for those institutes and the society in general?
Well, as you know, we have a president of the Max Planck Society. The president is very much a political position. So he interacts with the German government, with the German chancellor, with all the high-ups. And of course, he has the last, or she has the last say. But the day-to-day work and interaction directly with the institutes, that falls on the vice president to a large extent. We are three. We have three sections, three vice presidents. So I was the vice president of the biomedical section. And in this function, you handle the evaluations of all of the. So I had 30 institutes, 29 in Germany and one here, and you take care of a lot of the day-to-day. How should I say the function? You are in charge of the functionality of those places, right? And honestly, we have through history selected people for scientific excellence, and then sometimes it doesn't go along with excellent leadership. So sometimes you, as vice president, have to step in, and sometimes I saw myself as, if you have seen Pulp Fiction, Mr. Wolf, who has to come and clean up the car.
Yeah.
and that was sometimes my function. Other functions were I would say 90% very pleasurable, and like coming here in this was to try to figure out for me, in my head, it was to figure out a way to keep this institute on course and financed and allowing the excellent scientists here to keep on doing their science. And that's what we worked on in those days.
Were there any unique challenges that are faced by Max Planck Florida compared to other institutions in Germany that you were surprised by, or the fact that we're not in Germany? I mean, we're subject to a lot of specific rules and regulations that maybe are secondary to the Max Planck Society, but just imposed by governments locally and things like that. Were there any surprise challenges that came along that you had fun dealing with along the way?
Well, to me, they weren't a real surprise, but because if you look at our position here, it turned out that we needed heavy support from Germany to keep going, right? And then it's not a big surprise that the German taxpayers need a really good reason to ship 20 million euros per year over the ocean to the richest county in U.S. so that's, I mean, it's not so strange that we needed a really good reason, and we had to come up with that, and I think we did, right? Because first on the scientific side, I was always totally convinced. I have my colleagues here, right? But we also brought in 2 Nobel Prize winners, and we brought in another guy who is that caliber as well. And they very honestly looked through the whole institute from top to bottom, right? And they came up with an excellent evaluation in the end. So that made it possible to go to the representative of the German taxpayer, that is the government, right? And to say that here we have an excellent example of a German science embassy in US, and it's working really well, and we should really support it. And in this way we could really go on.
Maybe I could ask sort of about the broader question, which is sort of the, a push for internationalization of the Max Planck Society. Is that something that you see or saw when you were a vice president as a major focus or a goal? And why, if it is, why did you feel strongly about that?
This has 2 different aspects to it. One is, should we have more institutes over the world? No, I don't think so. I think this will be the unique, keep on being the unique institute. Should we be international? Yes, for sure. Like all scientific organizations, and we have the possibility of having Max Planck centers, which is the next level down from institute, and they're time limited. Sorry, and they give you the opportunity to interact very strongly, and we have one, for instance, between my institute and Sweden, my home country, where we get extra support of doing these things. But it's of course a big step down from creating an institute. And I think that when you have an organization that is, as we are, purely paid by taxpayers' money, it is hard to justify many of this. I think this is an outstanding example, but I think it would be hard to justify for German politicians to do more of them. We should concentrate on this one and keep it running in the very best way.
So you mentioned your home country of Sweden. You did your PhD work and your undergraduate work in Sweden. You did a postdoc in Arizona before deciding to go to Germany and start as a scientist in the Max Planck Society. How did that sort of international background influence your approach to science working with the Max Planck Society? Do you feel like there's anything as an asset, somebody coming outside the German science system that really brought a unique perspective to the table? And also, having been immersed in the Max Planck Society for so long, are there any things that you've learned along the way that you feel like are a model for how science should be done in general in an international context?
That's another 2 hours. No, but I think that it's hard to look on yourself from the outside, right? Mm-hmm. But the president that I was working with, he told me several times that I came up with views that he would never had as German, right? So, for instance, I introduced an onboarding for new directors that come into the society. Most Germans know what it's about, but when you come in as a foreigner, you really have no idea.
So it's sort of like an orientation to...
Yeah, it's a few days, and then I also took the initiative of writing the travel guide for the Max Planck Society, which is really like a Berlitz guide. It ended up 150 pages, and it fits in your pocket. And there is any information you want or need about Germany, about the society, about what it's like. There is 1,000 quotes of directors. We interviewed many directors to get their point of view. What was the problem of coming to Germany? What was the great thing, and so on. So to provide a perspective for those who come in the next generation of directors, because often when we, our system that we hire people, we court the people, we provide everything, we put millions in your lap as startup money and so on. But we also very rarely say, what do we expect? What do we expect of you as the highest level scientific leader in our society? We expect you to be a good leader. We expect you to follow decent rules and so on and so forth.
To be a role model.
To be a role model, to be a mentor, to be a supervisor. And all of these things, we take them as granted, but some people don't. So, and therefore also under our tenure, we started with having psychological discussions. We have people doing profiling of people that we hire, because we hire people for life. So it's a pretty important choice that
it's
you
kind
do.
of like recruiting for the FBI or something in the U.S. Like, you got to make sure that this
Yeah.
person's.
But think about my career, right? The society invests about $100 million into me during my career. That's
quite an investment.
Heck of a lot of money, right?
Yeah.
So you want to be sure that you invest them in the best way, in the best people. And we are very good at judging science. Now we are also good at judging that along with the science come also a decent person who can lead young people into their careers, which I always saw as extremely important.
You know, we recently had Patrick Kramer, who's the current president of the Max Planck Society, on this podcast. And one thing that he emphasized was not just at the level of scientific directors, but the importance of excellence across the board, from administration to scientific support staff to scientific directors, something that's like very intrinsic to the model of the Max Planck Society. You mentioned the sort of the evaluation process of, you know, if you're managing 30 institutes and you're in the biomedical section, presumably you're in kind of a constant rotation of going through scientific advisory boards, and which we just went through as an institute. On top of that, you're also running your own research enterprise of your own scientific work. So how do you find the time to balance these two essentially parallel careers, one where you're, you know, basically enforcing the mandate of excellence in science for the Max Planck Society, and two, also being an accomplished scientist? Sounds like a challenge.
It's hard. For me, ever since I started doing my own group as a scientist, I always wanted to have a group of people which had several layers, so that I had young scientists that were somewhat accomplished in their career, that worked together with me, that I formed sort of a leadership group of the department, and then there were postdoc students, of course, and so on. But thanks to this structure, I could keep pretty much on top of things also while constantly traveling. Because as you said, I had to do 15 evaluations per year, give or take. And each one of those took a week about. And then all of the other things, the group leader selections and one after the other. And then the critical cases and one after the other. So I was very seldom at home. But then thanks to having these 5 group leaders that I have working with me in my department very closely, they kept things running on the daily basis. And then I got home often in the weekend. We had the meetings and looking at how everything developed, discussing ideas and all the publications and so on. So in this way, if going back to what we said before, that you should be a good leader and so on. If I had been the old type hierarchical leader, it wouldn't have worked.
If you wouldn't have had like a team at sort of a faculty level.
If we hadn't been a tightly knit team that all cared about the department, it has always been. I have, for instance, obligatory coffee breaks. It's a Swedish. There is a Swedish noun and verb called fika, which means to have a coffee break and to take a coffee break. And this is sort of in the Swedish soul. And that's where you meet and you discuss, and 80% of the time you're just talking about a movie you watched or something, but 20% that's where the new
insights come,
right? So having these, and then monthly breakfast, department meetings, and so on. So in this way, the department, there is cohesion, and everyone knows what's going on, and so on. And I also always told them a little bit what I was doing as vice president, so they just didn't think I was
just proving that you were very busy.
So it's in this way it worked out, and we kept production up, and kept the ideas flowing, and so on. So it's possible, but of course I would have produced more scientifically if I hadn't done the vice president, for sure. But at the same time, it provided me with a really unique insight into how the Max Planck Society works, right? I knew, during those days, I knew every director of the biomedical section, 100. I knew the personality, I knew the weaknesses, I knew the strengths. I knew every institute, what was going well, what was not going so well, and so on. So you really, like a spider in the web. And then the unique thing with the vice president compared to Max Planck has several different layers. So we also have section heads, but the vice president has money in his pocket. So you can really, if you see colleagues that are at a breakthrough, for instance, then you can say, okay, here is a few million extra, so that you can really accomplish that extra step to go on, and so on. And that's also a really nice feeling, that you can really push your colleagues and help them out.
That's great.
So I wonder, now that you're back full-time, mostly focused on your research, is there anything that you sort of learned or took from your days as vice president that's influenced how you're approaching either your group or your research now?
Well, you can imagine I learned so much in so wide fields of biology and medicine and chemistry. Listening, I mean, I was sitting in to all of these evaluations where all the top people in the world present, showing their best ideas without restraint, and really wanting to make a good impression on those boards, right? So of course it affected. It widened my scientific scope enormously, and I can have. I was just thinking today. I was sitting with Rioje up here, and he was describing his intricate mechanisms that he's looking at. But I could understand part of that because I had been with Kai Johnson in Heidelberg, who is doing similar things and developing these different chemical markers and so on, and molecular markers, so no, definitely it gave me a much better vantage point. And also, I think in age, when I go on for a few more years, but then you will retire in the end, and then I think it will be amazing to have this scope, and you can start synthesizing things and getting a much better overview of things. So it was a gift at the same time as it was a hell of a work. Yeah.
So speaking of gifts, you gave us an incredible lecture today on the locust olfactory system, and we have to talk about it because just the intricate interplay of really precise, identification of the signals that sensory systems are using, the transduction of that signal, and then how that translates really concretely into specific behaviors is rarely demonstrated with such precision as it seems to be in this invertebrate system, which is something that I think Lesley and I, neither of us have much of a background working in, invertebrate brains. I don't think you've worked in locust. I certainly haven't. What was it like, sort of as you were sort of starting to assemble your research program as an early investigator, what drove you to invertebrate sensory processing, and also, how did you get involved in the locust as a model organism initially? What was the hook for you?
Two different questions, right? So the first one is serendipity, because I'm an ecologist by training, and I was studying animal ecology in the final years of my bachelor in those days, and I was convinced I was going to work on badgers or elephants or some
charismatic megafauna.
Yeah, exactly. I was totally into this, going to Africa. And then there was a graduate student position opening. There was a professor giving a very nice lecture about insect pheromones, and I was fascinated by it. So I went and talked to him, and then I got the position, and that's how it all started. I wasn't at all an entomologist before. I was a birder and looking at mammals and birds, but that's the way it went. And then with the locust, well, that came maybe because I still had this Africa tendency. So in the early 90s, I got the chance to work at an institute in Nairobi, and then I doubled up as safari guide. So I got to do both, right? So I usually did a month of locust work at the ISIPE, International Center for Insect Physiology and Ecology, and then I did 2-week safari for a Swedish safari company, taking people around in Kenya and Tanzania and showing them the fauna. Yeah. So that's the way it came about. Then I worked on locust maybe for 5, 6, 7 years. Then we sort of dropped it because, for different reasons. There wasn't funding. I had funding from a Swedish development agency, and we didn't get that funding. And then so slowly I moved to Germany as well, and then I left that project in Sweden. But then now we always kept it in the back of our minds that they had this. I showed you this strange evolutionary progression that they go from a normal architecture into this very weird locust style architecture, and that was always we should return and have a look at it. And then we have in the Max Planck Society, we have something called roundtable discussions with Chinese colleagues every year in Beijing or Shanghai. And as your vice president, you're allowed to set the agenda of one of those. And I set the agenda for animal behavior in general. And then we had the best Chinese and the best Max Planckers coming together. And then after that, extra money was announced that you could apply for. And then I went together with my Chinese colleague in Beijing, and then we got some extra support from both sides to look at the locust.
So some of the things you showed today included characterization of the response properties of like individual types of receptors in the locust olfactory system. So basically, coming up with a characterization of all the different odorants that they might be naturally prone to be interested in, and then when they interact with the nervous system, recording basically response profiles of the diversity of different odorants that an individual receptor might respond to. And what you showed in your talk today was that one, by and large, the olfactory receptors in the locust are very sharply tuned to very specific odorants. And then there are also a number of odorants that are, ethologically relevant to those species that indicate something about food sources or non-food sources, as well as information about their own species and their counterparts, other locusts in the large group of locusts. The thing that really made me laugh when you were talking was, you see these large hordes of locusts in nature before they've reached the flying stage of their development, mowing down, fields of grass, but they're basically scavenging for food. And if they slow down, they're going to get eaten by their friends. Mm-hmm. It's like this natural thing that you can probably observe in the wild if you have enough time and a camera, that the locusts that are stopping are getting gobbled up by the locusts that are coming up behind them. So it keeps the pack moving. And you also started to want to understand, like, what is the mechanism by which, a locust chooses to or doesn't choose to eat its friends? And the answer to that seems to be in the olfactory system. Can you tell us a little bit about how you went about trying to solve for the signaling that either attracts or repels a locust from eating its friend? Because it seems like incredibly intrinsically important to the overall, superorganism of the swarm, but also really finely controlled at the level of just individual proteins being expressed in the locust's brain. So how did you set about trying to solve how that happens?
So as I showed you, we had ideas that cannibalism is occurring, and then we did experiments where we crowded them more or less, and then we showed that cannibalism is a function of crowding. So the more crowded you are, it's pretty natural. But then we had when we crowded these nymphs that cannot fly, that can only walk, that get eaten from behind, then we saw one compound popping up that we didn't see otherwise, and that was this phenylacetonitrile.
PAN.
PAN. And this, the PAN is a usual suspect for locust. It's known from before to be involved in negative interactions to some extent. So we thought, that might be important, right?
So actually, before we get into that, can you tell a little bit about how you're able to tell like what types of odorants are attractive or repulsive or instigate some kind of negative interaction?
Yeah, sure.
Like what are some of the assays that you would use to assess that?
So then I mean, it's important to understand how important chemistry is.
Right.
Because we spent more than a year just getting the chemistry down. So we analyzed the odors emanating from different types of locust, the stages, the phases, and so on. And from food object, from non-food object, from eggs, from feces, from the locust. And so we ended up with 204 odors, which is a totally unique library of potentially active odors, right? And all of those odors we used to scan the receptors. And then for 42 receptors, we found the specific stimuli. And for one of those, PAN was the compound, right? The ligand. So when we then saw PAN popping up when we crowded them, and we already had a receptor that was tuned to it, then let's look at this now and what that receptor means. And then as today we have access to CRISPR-Cas technology, thanks to the Max Planck Nobel Prize a couple of years back to Emmanuel Charpentier.
Friend of the show.
Yeah. Then we could take that receptor out. And then we could show that by doing that, we could really abolish the negative behavior that we saw otherwise. So that was the first clue that this really functions, right? But then we had another handle as well, which made it pretty unique because our Chinese colleagues had already identified the gene underpinning the production of this compound of the PAN. So then we could knock that one out. And then we could see that made the animals very vulnerable because they couldn't produce the protective agent. So we could attack the topic or the question from in a two-pronged movement, right? And by doing that, it's pretty cool that you can really show that it happens.
So one thing I wanted to sort of interrupt is you've basically taken, a lot of tools that are used very commonly in, neurobiology, usually in mammalian systems, and use them in the locust. Are there particular challenges to using the locust as a model, or is the technology transfer pretty straightforward?
No, and I mean we are using some technologies that are not so common. That, for instance, the technique we use where we put the receptors into the fly, because the fly we know so well, Drosophila, right? Mm-hmm. And we know every neuron, and we know there is a special strain of neurons that comes from Yale that the receptor is missing in one neuron. So you can just take any other receptor and insert it there genetically. That is a big advantage of this system, because one of the challenges with the locust compared to other insects is that it has these conglomerates of neurons. So you can never really record from one neuron. You will see 40, 50 neurons spiking wildly, and it's impossible to sort them really. So by taking each receptor and putting it into the fly, there is only one neuron, and you can pinpoint exactly what it's responding to, right? So that's a big advantage of this system.
So I hate to ask this type of question, because I feel like the neuroethological question is already interesting in and of itself. But one thing that this sort of cannibalism story makes me think about is humans a little bit. And, to what extent do you think simple changes like maybe even more broadly than the olfactory system, but possibly in the olfactory system of humans as well, how do organisms like vertebrates and mammals and things like that, how sensitive would they be to some of these simple manipulations, like knocking at an individual receptor and seeing, is this an area where we just simply don't know enough about how we process our own interspecies pheromones? Are there things that happen during aging that maybe change, our processing of sensory information and therefore change our interactions with our, conspecifics? Like, how much of this sort of principle of, direct gene to behavior linkage can be extrapolated to, animals with much larger brains and much more complex nervous systems?
So first, the unique thing that we already mentioned about the locust is that each neuron seems to be so exquisitely tuned, right? Yeah. So in our nose, we have about 400 receptors. And as far as we know, most of them are pretty widely tuned. So they respond to a whole spectrum, right? But a different spectrum. So this is the background that we can really smell millions of different odors with our 400 receptors, because they will be activated. I always say it's like playing a piano, right? You can play many tunes, especially if you have 400 keys to play on. So that's a principal difference. Then when we come to transferring the locust system to ours, what could happen? Well, the first thing you should know is that we are quite polymorphic in our setup of receptors. So there are certain that's how the Swedish Linnaeus he characterized different phenotypes by showing that certain people cannot smell certain odors. Mm-hmm. Like if you take androstenone, the pig pheromone. Mm-hmm. Some people, you open the vial in the room, they have to go home, right? While I don't smell it at all. I can put the vial up to my nose and inhale, and it's no problem, right?
Somewhere I'm sure that's a superpower.
So I mean, there is big polymorphism within among us, right? And so it's already there. Then, when it comes to the pheromone side or these interactions, we know too little, and we don't know the receptors and so on. So, I mean, this is more or less my talk for tomorrow at Science Meets Music. But if you look at an insect, at a moth, for instance, there is one receptor is so overexpressed in the male, and the antenna has grown into a huge thing, right? And it's only there. If that is targeted, then it only means one thing. There is a female up there, and I don't care about anything else anymore, right? So, and if you knock that one out, then it's over. He will never find a female. But for us, I'm always saying that we are so complicated organisms, right? We are multisensory for sure. We carry an emotional backpack that is often huge, memories, impressions. So I think that our system sort of makes up a little bit. If maybe if you have
There's some guardrails.
yeah, exactly. And if you have lost a little bit on the olfactory input side, maybe you make up by having a little bit more visual input when you choose your partner or something like that, right? So I think in that way it's always hard to compare to insects because they are often quite hardwired.
So before we let you go, I wanted to first mention that you recently wrote a book, Smelling to Survive, where you talk about, all of your incredible experiences looking at the sense of smell and how nature has shaped those smells and those processes. So I wonder if you could maybe share maybe a story from the book or one of your favorite experiences about an interesting olfaction.
Yeah.
Like what's the cocktail party thing that you bust out to just shock people or surprise people?
For my book?
Yeah.
Yeah, no, I like the chapter. I like the chapter on our field expeditions to Christmas Island to look at. So insects were on land for 400 million years, right? But crustaceans have only been up for 5 million years. So, and they went up 5 different times onto land during evolution. So we have the hermit crabs, we have the normal crabs, we have the crayfish, we have the wood lice, and we have the small things living on the beach. The gammarus in Latin. And the strange thing for these, of course, is that they all had to develop land smelling. It's not an easy thing, right? We don't think about it, but you live in land, in water, and then you have to go up and smell in air. So how do you solve it? So that was our main question. And then we started looking at this. And interestingly enough, one of these 5, the wood lice, they have totally dropped olfaction when they walked out of the water. So in their brain is just a big hole where the olfactory center used to be. 3 of them have more or less remained, the crayfish, the crabs, and the other one. They look very similar. But then the hermit crabs, their brain has absolutely exploded with olfactory tissue to an extent that they had to push their memory centers up into their eye stalks. and it's really amazing and such a packing of neurons. So we went to Christmas Island first really to look at these. They are the world's largest land-living arthropods. They go up to 5 kilos and they live 100 years. So they're called rubber crabs or coconut crabs in English. The pressure they have in their claws is double what we have in our jaw, which is the highest pressure we can exert. So we went out and then no one knew much about these guys at all. So we started following them by satellite to see could we figure out if there are females somewhere, males somewhere. We had experiences like David Attenborough, where I went down with rope into caves and found the females full of eggs sitting waiting for the right tidal season. And so that chapter I quite like. There is an anecdote in there where it's more taste than olfaction really. But we saw these crabs. They love a special kind of fruit that grows on an indigenous palm called arenga. And they can, by their olfaction, predict 2 weeks before these fruits start falling of the tree, the big males start gathering below the tree. So just before they start falling, there can be 50 of these guys, this big, sitting below the tree. And then we were watching them, and we saw them eating these fruit, right? And as a biologist, I said, okay, I try it as well. So I went and I took 2 of these fruits in my mouth, and at once my whole tongue swelled up.
Oh my God!
And I got severe breathing problems, and it turns out that they're really, really toxic, and so I was like falling down, and my wife was running around. What but then we got some water, and it slowly subsided, and then of course that shows the importance of looking carefully, right? Because then I looked again at the crabs, and what they did was that they carefully, with their claws, they peeled off the flesh, and then I cracked the seed, and in there was a very fatty, nice, non-toxic part of this fruit. And then we had a game warden with us who helped us out, and then he came up to my wife, and then he asked, how did he get this old?
That's amazing. For this story and many others, definitely check out the book. Dr. Bill Hansson, thank you so much for joining us on this podcast today. It's been a pleasure getting to know you and hearing about your amazing career in science.
Thanks for the invitation. It was great to be here.
Absolutely. As always, thank you for listening to Max Planck Florida's Neurotransmissions podcast.
If you enjoyed this episode and would like to help support the podcast, please subscribe and tell your friends.